Hello, this is Nitin Dahad with the Times, and I'm here on the show floor at Imec ITF World 2026, talking to Barun Dutta, who's a chief scientist at Imec. Barun, hello, how are you?
Hi, thanks, Nitin. Nice to meet you, Nitin.
So, Barun, on the keynote, the opening keynote at the conference here, Patrick talked about Neuropixel 3, and it's kind of like the merger of biology and science.
The way I would put it is where nanotechnology meets neuroscience.
Neuroscience, okay, yeah.
Okay.
So it's quite interesting because what I like to do is dig a little bit backwards into how you got here and what exactly this is doing. So just tell us a little bit about the timeline.
So I'm a semiconductor engineer, have been all my life. I'm in fact a wafer builder. I've worked in all parts of the semiconductor value chain, from epitaxy to material science to device engineering, and still continue to do that. That's a large part of my responsibility and job of programs. And actually, it's around about a decade ago, sometime around 2013, 2014, because of some of the challenges that were arising in the field of neuroscience, where people wanted, for the first time, were trying to take a more systems approach to understanding the brain, that they wanted much higher resolution, single neuron specific data, which was representative of circuit functionality in the brain. That's how they were building. That's why the term system neurons comes, because they were looking at trying to build, they're looking at a circuit picture of the brain, which starts aligning much more to with what we do. Is we try to understand systems from a circuit point of view. So there is some correlation, but there are different types of circuits. These are biological circuits. But what we realized in a lot of these conversations was that they had a tool at that time, which is largely a 2-photon microscope and optogenetics, where they transfect cells and look at optical responses. But they were limited to looking at cells close to the surface of the brain. Okay? But they were beginning to realize that actually the brain activity was very distributed and existed across the brain and across all depths and scales.
Got it.
Okay?
Yeah.
And they were looking for a tool. Obviously, this tool had to be minimally invasive, because basically introducing the tool does do damage to the cellular structure of the brain, and they wanted to minimize that. And I think what we came to the realization at Imec, and I think that was the unique realization, was that some of the very advanced methods of semiconductor fabrication that had developed in the sub-100 nanometer nodes, which were typically not accessible to any of the academic communities. Okay, if they could be engineered similarly to way people were engineering microelectromechanical systems in silicon and connecting them up with the advanced CMOS, we could potentially address this challenge. Okay, so this was basically the genesis of a new approach to building large-scale sensors for this field. Okay, and that is what, with long conversations with some of the leading scientists of the world, and actually there were 3 of them who stood out. One was John O'Keefe, who went on to win the Nobel Prize, and his 2 colleagues, Edward Moser in Norway, and the folks at HHMI Genelia, that's mainly led by Carl Saboda and Albert Lee, and Christoph Koch at the Allen Institute. They all knew the problem, and they recognized and appreciated that might be our approach could be a solution to what they were looking for. So it was a very collaborative effort, where they actually really did very good work in trying to define what would be a very good experiment to do, because we wanted to validate it. So you can do it. And what we went on to do is have a joint collaboration for the Neuropixels 1.0, which was a 5-year exercise of going from scratch.
And by the way, the results we see up there is with result of 1.0.
1.0, those are the results of 1.0. And essentially, basically, this 5-year exercise yielded Neuropixels 1.0, which was available to the scientific community in about 2019. And this was essentially the first time that you had a tool which could do large-scale mapping of brain activity.
Now, when you do, just quick, we need to understand the parameters. What are you looking at? The density, the cells.
So what we basically were able to do with a single cell, basically, where people at that time were recording something between single numbers of neurons and to up to maybe 10, 15 because the number of channels on an electrode was something between 16 and 32.
Okay.
But we
and that's not linked to the processors.
No, and we basically took it up by nearly an order of, but you could say a factor of 20, and actually provided coverage of over a 10 millimeter span. Okay, and you could introduce multiple of these probes because of the small form factor into the brain simultaneously. Okay. So essentially, what you suddenly had the ability to look at 3,000 to 5,000 neurons simultaneously performing a task.
And that's kind of what you're.
And this kicked off an international collaboration called the International Brain Lab, which was supported by the Simons Foundation and the Wellcome Trust, where about a group of labs got together to basically, for the first time, try to do a large-scale mapping of a activity response to this thing. And that was done, and it was a very successful project, which for the first time, people had a full brain activity map, functional activity map on a single task across multiple animals successfully.
And I just want to highlight that for the audience, because basically it's 139 mice. 459 sessions,
and it's over 600,000 neurons.
Which is
single, and that's where the density matters. That's where the density matters, because before this, and even today,
I'll just show this to the So this is the probe, the shank. And this is where you're integrating all the electrons.
This is a real digital device. The analog ends on the chip, and then it's fully digital. So the noise performance of all this was an order of magnitude better than anything else that was available. So we changed the entire, let's call it, to a space of how you did this experiment. In fact, that's why we put that it was a new electrophysiology had been around since Volta. Okay, but this was a new era, because you were, instead of recording one neuron, you were recording thousands, and now we're getting to the point of doing tens of thousands, and most likely with the 3.0s, we will be doing 100,000 neurons in a single animal and in a single session.
Now, just tell us a little bit about the electronics in that. So there's the MEMS, there's a CMOS.
This is an integrated MEMS device.
Okay.
Which has actually nano MEMS, because the structures are all down to, some of the dimensions are down to like 20 microns.
Okay.
They're very small, and the electrodes on them are like 10 by 10 microns, and each of them have a full high performance, some of the highest performance amplifiers in them integrated. Okay, and what is important to realize, unlike an imager which runs at 60 frames per second, these are running at 30 kilohertz. Okay, because they have the same type of resolution and sensitivity as a CMOS imager, but at much higher speed.
Right.
Okay. So, and we had to keep the power down, because they had to be low power. They cannot generate a lot of heat, because the brain cells don't like to be beyond their 40 degrees centigrade limit. Okay.
And then the other one is like the rejection as well, isn't it?
And so the question is, and they have to be of the dimensions such that you don't induce, because if you look at a human DBS electrodes, they have millimeter dimensions. They induce enormous immunological responses in the brain. These are all smaller than 100 microns. Okay, so they do induce immunology, but they're much more limited.
Got it.
Okay, and we see a clear roadmap that we do see a path beyond 3.0 to 4.0, or might be 5.0, which will be again increasing the density. And so we do believe that fundamentally today, people are reaching densities of recordings which the tool is no more the limitation to the science.
Now, let's move beyond the tech and the science. What are the real-world implications?
So the real-world implications that we are essentially enabling, let's call it, it's very early days. We're in the discovery process of discovering a new way to understand various, especially sensory responses of the brain. So across especially the visual system, the auditory system, and we've got some early data on the auditory system where we clearly see that we can actually, with this tool, in the areas of speech, in a human, really reconstruct very efficiently basically the spoken word.
So, for example, people with brain damage,
Brain-
the aphasia, praxis speech, and all that.
But the best example here is most of these patients that we look at either have had brain cancers or brainstem stroke,
Yeah.
and we are able to use that to the tool to reconstruct their speech. We have been doing this now for about 3 years, and we have published one of the first papers on this, where we can actually show that compared to whatever technology was available before, we today can pretty much reconstruct a person's speech to within about with 1%, less than 1% error.
And I don't want to raise hopes, because I mean, I'm really
No.
hopeful of my mum, because I feel like I lost my mum when she had a brain stroke, but because she couldn't communicate. But
yes.
I guess it's like
But
20 years.
Yeah, it's still a decade out. But
Yeah.
we believe we are building to build models where we fully understand what, how, and because one of these things is that it's very easy to get to a 50% accuracy, but in things like in areas like speech, that is not good enough.
Yeah.
Okay, because you get dead noises and dead centers and other. We are up to the levels where we actually think we can find solutions to people with these diseases.
One last aspect you mentioned to me is you're getting so much data, you could actually train models for individuals for whatever purpose it
So
might
that
be.
is that's a more recent realization, especially with what's happening has happened in the last 2 or 3 years in the AI field. What has happened in the AI field, you will hear this from many people, that they are also running out of data, and one of the things that the transformer models did was they improved they found a solution to this limited availability of data to train models. What we are looking at is these sort of behavioral data, which is based, because typically data that is used today is based on the outputs of humans.
Okay.
We potentially have the possibility to build models where we can do it with the inputs to humans and how the responses are.
Got it.
Okay,
Yeah.
so these are very early days, but there could be a new way of building models which are relevant, particularly to disease, because we could possibly build functional models of diseases.
Yeah.
And what is important is do some of these experiments for treatment in silico,
Got it. Yeah.
and then come back to the tissue to make sure that we have because this could be a very nice way to personalize a solution per individual.
Excellent. Well, Barun Dutta, that's been fascinating talking to you.
Thank
Thanks
you very
so
much. Nice
Thank
meeting
you.
you.